The mission of the Molecular Imaging Innovations Institute, MI3, is to develop, implement, and translate a wide variety of probes or chemical agents for improving the diagnosis of disease and the treatment of patients. This spans both a wide range of diseases: cancer, central nervous system disorders, inflammatory, infectious disease. We not only make a number of these probes, but we also use them for a wide variety of diseases to better understand them and to understand the processes that influence these diseases so we can intervene with appropriate therapies for the patient. We have to work with the right team of people in the right fields for the given questions we're trying to answer: chemistry, physics, biology, immunology, pathology, computational sciences. We must have a firm understanding of these fields because they're influencing the responses we're measuring. You can almost see that without all these fields, you would have an incomplete answer. This is all very important, and we've incorporated over time many different fields. So in nanomedicine, for example, we develop small molecule drugs and peptides and antibodies, nanomedicines, and cellular engineering products that can also work together to achieve the goals of improving outcomes for patients in the end.
We develop on-demand nanofiber, displaying organ-specific targeting and/or retention property to improve the safety and efficacy of chemotherapeutic treatment. We can specifically carry the drug to tumor site with a minimum of target accumulation in other organs in the body, and we can also return on site to after release a broad spectrum of anti-tumor activity over time for a more specific long-term treatment. So there is an urgent need to develop new weapons to fight cancer, and I think nanofiber may have a great potential to ensure this mission.
It has now become clear that the physical chemical properties, such as the size, the shape, the surface charge of a drug or its carrier, affect their in vivo trafficking. For example, using PET, we were able to identify certain particles of 2 dimensions with negative charges are more likely to bypass the physiological barriers, reach the tumor and penetrate the tissues and stay at the tumor site.
The outcome of all of this is to improve patient care and to have better tools to answer a lot of the questions that often arise in the care of the patient. So by designing probes that are sensitive to that, we can really make earlier diagnoses and predictions to help guide the care of the patient in the future.
Conventional radioimmunotherapy didn't have a sufficient safety window to be effective. So we use a multi-step approach in order to physically separate the antibody targeting step from the radionuclide payload application. It's a way to increase the tumor to normal tissue targeting ratios of your therapy.
Targeting immune checkpoints is currently dominated with antibody-based therapies, and this comes with many limitations. So small molecules have the advantage of addressing many of these limitations, such as potential for oral administration, such as minimizing the risk of immunogenic responses, more efficient tissue penetration, as well as a lower cost of production. So what excites me about my research is the potential to maximize the body's immune response against cancer and other diseases. By making novel small molecule immunomodulators, we have the opportunity to develop more efficient therapeutics that can have potentially lower side effects.
PET is, I think, really an outstanding tool for understanding the mechanistic basis for disease, and that's because imaging really can inform so much about how we view medicine and we view treatment. We can see the whole body at one time, and that allows us really, I think, to diagnose disease more accurately, to understand the implications of treatment, and really accomplish what I think is the long-sought goal of personalized medicine.
So we are extending our MRI method by integrating with optical probes that can provide valuable additional information about the tumor cells, such as therapy-induced senescence or a change of pH in tumor microenvironment. The synergy between MRI and these probes provides a unique opportunity for us to better understand how tumor respond to therapy and the underlying mechanism. I think our institute has many talented researchers with a diverse background who will shape the future imaging science, clinical radiology practice, and medicine. And it's really exciting to work with all of them daily.
The Institute exists to achieve all of these goals. It makes a number of products that are aimed at the clinic to do a better job than the current probes and to overcome the obstacles that have plagued current probes.